Thank you for joining us for Metabolon's half-hour webinar series, where our topic today is Metabolomics and Lipid Profiling of Skin. Your presenter for today is Dr. Fanny Tang. Fanny holds a PhD in Nutritional Biochemistry from the University of North Carolina at Chapel Hill. She joined Metabolon in 2014 and is Senior Study Director, where she is responsible for analyzing and interpreting large metabolomic data coming from various industries with a focus on applied markets, which include human and animal health and nutrition, consumer goods, and skincare. I'll be turning the presentation over to Dr. Tang now.
Good morning. Thank you for calling in today. Today I'll be talking about metabolomic and lipidomic profiling of skin. The audience today may have different levels of experience or exposure to skin research. With that in mind, I will start the webinar with a short primer on skin biology. I will then walk you through the different offerings that Metabolon has that's pertaining to skin research, and that includes the global metabolomic platform and various lipid panels. The utilization of this technology can also be exemplified by several case studies completed by our previous clients. So skin is the largest organ in our body, and it has several important functions, including protection, regulation, sensation, homeostasis. As an organ of protection, the skin forms a barrier that protects us from mechanical impact and desiccation. It also prevents the invasion of microorganisms as well as UV radiation or chemical exposures. As an organ of regulation, the skin can regulate body fluid or temperature via hair and sweat, and skin also serves as a reservoir for vitamin D synthesis. And as an organ of sensation, the skin has a very complex network of nerves that allow us to feel the environment, including the heat, cold, touch, and pain. And since the skin can excrete, absorb, produce, and store metabolites, the skin can also reflect the whole body homeostasis. Those functions of the skin are carried out by its complex structure, and the skin has two major parts, the epidermis and dermis. The dermis is where all those appendages locate, so things like blood vessels, sebaceous gland, sweat glands, nerves, all locate in this compartment. The epidermis is the most outer layer of the skin, and it is predominantly composed of keratinocytes. So the keratinocytes start at the stratum basal level, and they keep proliferate and migrate upwards. Eventually, those keratinocytes become a nucleus, corneocytes embedded in a lipid matrix. This whole area is called the stratum corneum, which provides most of the barrier function of the skin. And let us not forget about sebum, which is the oily substances produced by the sebaceous gland. Although sebum is often connected to acne, sebum itself has protective effect. It helps repel the water so that it prevents excessive skin hydration. It also helps lubricate the hair and the skin. Importantly, those structures all are composed of metabolites. So, for example, the corneocytes in stratum corneum has a lot of amino acids, which function as the natural moisturizing factors, reabsorb water into the cells to prevent dehydration. Both stratum corneum and sebum also have very unique lipid composition, which we will talk about later on. But those metabolites are not only the structure components of the skin, but also the main driver behind those skin functions. Importantly, those metabolites are also very sensitive to changes, and they can be influenced by factors such as the diet that you eat, the environment that you're exposed to, whether you smoke or not, what bacteria live on your skin. So really, the measurements of those metabolites serve as a summation of all the factors that drive the final phenotype and provide a readout of the final physiological state. And that's what we are interested in. And Metabolon has different technology that can be used to measure those metabolites. And the approaches are dictated by the study objective. So if an investigator is unclear about what metabolite they want to measure, the investigator can start with the metabolomics alone or in combination with lipidomics. If the investigator knows what metabolite they want to measure, Metabolon also have a targeted assay team that can establish the assay for the specific metabolite in a specific matrix. So let us start with the general metabolomics. What you're looking at here is a wall chart of those small molecules in our system, and each of the blue dots represent the biochemical that we can detect that are found in our library, just to show you our coverage. Essentially, what Metabolon does is that we take the samples from the clients, and when it comes to the skin research, we have a lot of experience working with the D-squam tape that is used to collect Stratum corneum. We can work with cells like keratinocytes. We can work with skin biopsies and even 3D cultures such as the reconstructive human epidermis. And what we do is we monitor and screen across all of those metabolites in the given sample and report those data back to you. Not only we monitor the metabolite that you may be interested in, for example, the amino acids, we can also monitor metabolite that you may previously not anticipate. So this unbiased global approach can really provide a lot of insight to your research questions. Now, the challenges of wanting to monitor those metabolites is that they have very different properties. So in order to cast a wide net to capture many of those, Metabolon has employed 4 different mass spectrometry methods. And we have several publications on our technology. If you're interested, we're happy to share those with you. But in short, each of those methods generate a large amount of ion features. Those do not mean there are hundreds and thousands of metabolites. Those are just fragments of the metabolites. So the challenge in the world of metabolomics is really how to handle this amount of information. And over the years, Metabolon has established our own software that allow us to eliminate the noise and reduce the data down to signals that are really biologically meaningful. And at the heart of our technology is our library-based approach or chemocentric approach. We have a library of 4,000 named biochemicals and a library of 7,000 to 8,000 unnamed biochemicals. Each of those biochemicals have very unique characteristics, including retention time, actual mass, and fragmentation patterns. So by querying those characteristics, those signals from the client samples to our library, we can get really accurate determination of each metabolite. Once the metabolite lists are determined, we can then perform statistical analysis that is appropriate for a given study based on the study design. And our PhD level study directors can interpret the data so we can start understand what metabolites change and what does that mean to your research questions. Now, over the years, we have completed more than 5,000 studies, and that has led to more than 800 publications. So really, there's a lot of knowledge coming out from our work. And that is a very quick rundown of what we do when it comes to metabolomics. Next, I want to talk a little bit about lipidomics. And Metabolon actually has 3 separate lipid panels, including complex lipid panel, sebum lipid panel, and Stratum corneum lipid panel, and I will talk about each one briefly. So complex lipids are lipids that usually have a hydrophilic head group and 1 or 2 more of those hydrophilic fatty acids. So for example, here we're looking at a phospholipid that has a choline, phosphate, glycerol head group and backbone, and many different type of fatty acids. So the different combination of the head group and the fatty acid can generate hundreds and thousands of unique lipid species. So what the lipidomics does is that not only we want to monitor those complex lipid, those head groups, but also the fatty acids within those lipid classes. And by summing up the fatty acids within a lipid class, that will yield a lipid class concentration. And by summing up a fatty acid across all the lipid classes, that will yield a fatty acid concentration. So once we get the concentration data, we are able to calculate the composition as well, so we can understand within the same concentration what is the contribution of a specific lipid species. And We have a complex lipid panel, and this lipid panel uses a select ion DMS technology that we co-established with AB-Science. With this technology, basically we're moving away from the traditional TLC scraping separating those lipid classes. This technology allows the passage of a specific complex lipid class to pass at a given time by adjusting the voltage. The complex lipid then goes through what we call the multiple reaction monitoring, and the intact mass and fragmentations are monitored here so we can know what are the fatty acid species within those lipid classes. With this new technology, we are able to monitor more than 1,000 individual lipid species across multiple different classes, including phospholipids such as PC, PE, PI, and their lysoforms. We can monitor sphingolipids like sphingomyelin and ceramides, as well as neutral lipids including triacylglycerol, diacylglycerol, monoglycerol, free fatty acid, and cholesterol esters. Naturally, this complex lipid panel is designed for tissue types that have those lipid classes. So things like plasma, serum, most of the tissue, skin biopsy will probably be run on this lipid panel. We have a separate lipid panel for the sebum because if we look at the lipid composition of sebum, they have very different composition. The sebum is composed predominantly of triacylglycerol, free fatty acid, wax ester, and squalene. So if we run the sebum sample on the complex lipid panel, we will be missing out on information such as squalene and wax ester because those are not monitored in the complex lipid panel. And that is the main reason why we have a specific panel for sebum. So with this technology, we are able to monitor close to 1,000 individual lipids. And once again, most of those lipids come from triacylglycerol, wax ester, and of course for squalene, there is only one possible combination. Importantly, the mRNA does not distinguish the isomers of fatty acids. And as we know that sebum has a very unique fatty acid, a lot of those are branched chain fatty acids that have iso or anti-iso composition. Therefore, a subset of the samples was actually taken out and run through what we call the GCMS FAME analysis. With this second analysis, we are able to report back the absolute concentration of 32 fatty acid species. Those are total fatty acid, meaning that it's the fatty acid from across all of the lipid classes. And this assay is optimized for sebum that is collected using the sebutase and is optimized for humans. Similarly, we have a lipid panel for Stratum corneum, and this is because the Stratum corneum once again have a very unique composition, including cholesterol, ceramides, various forms of ceramides, and free fatty acids. So naturally, the Stratum corneum lipid panel is designed to capture those lipid species. And this panel is optimized for the Stratum corneum that is collected using the D-squam tapes. And once again, this is optimized for human as well. So I just want to remind all of you that while there is specific lipid panel for different matrices depending on the lipid composition, all of those sample types can be run on the global metabolomic platform. And the global metabolomic platform can provide you information on amino acid, peptides, carbohydrates, energy, nucleotide, cofactors, xenobiotics. So really, the combination of the metabolomics and the lipidomic platform can provide the most comprehensive survey of the metabolome. And just a little bit about the sample collection for sebum and Stratum corneum. The collections are pretty simple and non-invasive. For sebum, basically what you would do is, for the participant, you will clean usually the forehead with the alcohol. You will apply 2 sebutases side by side, wait for 30 minutes. So what we're measuring are the amount of sebum produced during this time frame. You can then store the tapes in Sebum tape storage carts and then put it in a Ziploc bag and then send it to Metabolon. And this is a nice way to save freezer because you can stack the sample on top of each other. For the Stratum Corneum, similarly, we use a D-squam disc. Instead of putting the tape side by side like the way you would do with the Sebum tape, what you do is you put one tape down, peel it off, put another tape down, peel it off, and repeat it 4 times. And the tapes can be stored in the storage carts. And some investigators also found to be very convenient just to put the tapes in cryo-vial and ship those in freezer box. Either way will work. In terms of sample amount, if you're just doing the lipid panel, we ask for 2 Sebum tapes and 4 D-squam discs. If you want to do both the lipidomic and metabolomic, we ask for double amount of the sample. So basically 4 Sebum tapes and 8 to 12 of the Stratum Corneum D-squam discs. So I just want to summarize everything up to this point. Skin is a very complex organ, and Metabolon has a lot of experience working with different skin-related tissues, including sebum, stratum corneum, tissue like skin punch or 3D culture, and we can also work with cells and media as well. For all of those matrix types, we can do the global metabolomics, and that's what we call the HD4 here, which is the 4th generation of the platform. And depending on the lipid composition of those matrix types, we also have 3 different lipid panels that you can select from to best cover those complex lipids in those matrix types. And those technology has many applications in cosmetic and skincare, and some of those can be to answer very basic research questions. For example, what is the impact of smoking or aging? It can also be used for product development to assess treatment efficacy. What does this treatment do to the skin biology? And some of the data may even be used for establishing or support claims. The technology can also be used to evaluate the mode of action of specific ingredients or even to monitor potential toxicity issues. So with the remaining time, I just want to give you 4 really high-level examples of how others have applied those technology to their research questions. And hopefully this can also help you understand how you may design your studies as well. So the first example aims to address a very basic question, and that is, what is the impact of UV exposure? So to address this question, the investigator collects skin biopsies from Caucasian females, and the samples were collected from inner arms and outer arms. Those represent the skin protected, the sun protected and sun exposed skin. And we have 25 samples per group. And this is very important because humans tend to have a lot more biological variation, and we really want to have a higher number to make sure we can cover or detect the differences between the experimental groups. And those skin biopsies were run on the global metabolomic platform. And if we look at the principal component analysis, we can see great separation between the sun-protected and sun-exposed skin, suggesting that the UV exposure does have a great impact on the overall metabolomic profile. And if we look at the specific pathways, we see that glutathione, which is the major antioxidant in our system, reduced in the UV group, and other markers of oxidative stress, including other metabolites in the glutathione pathway, as well as metabolites in the purine catabolism, such as xanthine urate, were all significantly elevated in the UV-exposed group. Collectively, all of those suggest that the sun-exposed skin is under high oxidative stress, and some of those metabolites can be used as a potential target or biomarkers for UV exposure, as well as to gauging or testing drug or treatment efficacy. And the technology can also be used to understand for product development. So in this study, the subjects received no treatment or treatment of a new formulation. And samples were collected at pre or post time points. Once again, we have around 25 individuals per group. And for this study, instead of biopsy, the stratum corneum were collected using the D-scrum tapes and ran on the global metabolomics. The study director will interpret the data, and the deliverables from the global metabolomics include the statistical heat map, biochemical pathways, as well as interpretation. And for this one example, our collaborator investigator was able to use metabolomics to better understand the benefit of a product. And let me just read this to you. This is from our collaborator, L'Oreal, who says, "What is metabolomics? A science of the infinitely tiny that can help us analyze cell activity in real time. Thanks to metabolomics, Lancome researchers have been able to help the skin to function more deeply and improve its reactions to external stresses." The past two examples are on the global metabolomics, and the next two examples I want to share with you are using the sebum lipid panel. So for this specific study, the investigator is interested in what is the impact of acne versus no acne when it comes to sebum lipid production, and what is the, with the people with acne, what is the impact with topical and systemic treatment. And what they found is that people who have moderate acne compared to, they have elevated fatty lipid composition, including wax ester, GAG, TAG, and squalene, compared to individuals that do not have acne. And this is consistent with the literature that individuals who have acne tend to have more sebum production. And when it comes to the treatment effect, the investigator was able to find that although the topical retinoid treatment did not have significant impact on the sebum lipid composition or sebum production, the systemic retinoid was able to reduce all of the major lipid classes in the sebum as well as the total sebum production. So overall, they found that with the systemic treatment, they are able to reduce sebum production, and this study was presented in a conference as a poster in 2015. Finally, the technology can also be used not only in the cosmetic or bioapplied market, it can also have many utilization across different intervention and diseases. So for this example, this investigator is working on a drug called TVB2640, which is an inhibitor for fatty acid synthesis. And this drug is used for cancer patients because a lot of the cancer cells use a lot of fatty acid for energy and growth. So the idea is that if this drug can impact fatty acid synthesis, does that also impact the fatty acid composition of sebum? And can sebum be used as a non-invasive means of drug target engagement? So for this study, individuals with cancers went through the monotherapy of TVB2640 or combination therapy of TVB2640 with another drug for many days. And sebum were collected using sebum tapes, and that was run on the sebum lipid panel. Each of the section of the bars represent the samples from the same individual across different time points. As you can see, there's a lot of individual differences. But within each individual, the amount of sapienic acid as well as the short-chain fatty acid and monounsaturated fatty acids reduced dramatically after the use of the monotherapy, suggesting that with this drug, it also impacts the fatty acid synthesis in the sebum. And when they look at the triacylglycerol composition, the triacylglycerol levels also reduce within those subjects, either with the monotherapy or the combination therapy. So in conclusion, this drug results in reduced sebum production and results in some of the fatty acid synthesis in the sebum as well. And the sebum analysis can provide a non-invasive means of assessing this drug and potentially serve as a non-invasive way to test the drug efficacy in those participants. So in conclusion, the skin is a very complex organ that has important local and systemic functions. They are central to, the metabolites are central to much of the skin functions, and Metabolon has a wide range of capabilities and methods for profiling the skin. And those can be applied for dermatology research and development, but it can also be used for biomarker research across a variety of disease and interventions. Thank you for your time, and I really appreciate you calling in today. With that, I'm happy to take any questions. Okay. So the first question is, what is the role of omega-3 and omega-6 in skin? So as we know, omega-3 and 6 fatty acids are essential fatty acids, so they're certainly very important. And especially the linoleic acid, which is omega-6, is the most abundant PUFA in the epidermis. So naturally, those fatty acids contribute to the barrier function. In addition, since those fatty acids are also precursors to cytokines, eicosanoids, so they also have roles in regulating inflammation. And in addition, there are also other suggestions that those fatty acids may be involved in promoting wound healing and even UV protection. So I think there are a lot of functions for those essential fatty acids. So the next question is, skin microbes are known to affect the skin function in many ways. Are you aware of any study where skin microbiome metabolism being studied, or is there any way to look at microbiome versus host metabolites? So I think in the recent publication, there is one publication that I remember suggesting that microbes can impact vitamin B12 synthesis in the host. That work was not done by Metabolon, but that paper certainly suggests there is important role in microbes when it comes to skin health. Now the question is, how can we distinguish metabolites coming from microbes or coming from the host? So when we get the skin samples, since the metabolite can be produced from either microbe and host, so for most metabolites, some of those are probably contributed by both. But we do know from our experience there are certain metabolites that are exclusively produced by microbes, and because humans just don't have those enzymes for it. For example, a lot of derivatives coming from aromatic amino acids, once we see those metabolites, we know those are exclusively coming from microbes. But there are other metabolites like lactate, which can be contributed by both the host and microbes. Of course, then we can't say exclusively it's coming from microbes. But from our experience, we know, what is coming from the bacteria and what can be contributed by both. So by doing the global metabolomic platform, I think it is very essential because not only it provides you the information on microbial-derived metabolites, it can provide you a sense of the host metabolism, and it can also provide information on diet and xenobiotics, so you can start understand how the microbe can interact with all those other host or xenobiotic metabolism. So the question is, how can you detect collagen or elastins in the skin? So the metabolomic platform is designed to capture small molecules. So collagens, elastin, those are larger molecules that are too big for the platform. But from the Stratum Corneum tapes, these one tapes, we do regularly see a lot of the building blocks of collagen. For example, hydroxyproline, proline, prohydroxypro, and those can give us indication of this collagen turnover. So do you run studies compare treatment effects using 3D tissues or in vivo samples? So what we do is that we treat each samples independently. So basically, the 3D culture will be run together and the in vivo sample will be run together. So we cannot compare the data across matrices, but we can compare data within the sample set. So for example, what we can do is for both tissue type, we may have a control and treatment group, and we can see, can we see the similar findings, similar change by the treatment that is consistent observed between the two matrix type. But we cannot compare glucose from 3D culture to the glucose from a cell culture, for example, if that makes sense to you. Another question is, what is the level of quantification? So it's for the small metabolite platform, it provides data in relative quantification. The lipid panels provide concentration data. So it is quantitative in the sense that for each lipid class, there is at least one internal standard that is used to calculate a concentration. So the short answer is that all the data are quantitative. For the small metabolite platform, it's relative quantification. For the lipid panel, it's concentration.
Thank you for attending our webinar.